Good afternoon everyone. I am Ganesh.Tilve, Assistant Professor in Department of Biotechnology. Recently we had an faculty enhancement program based on different bioinformatics tools. Today I am here to explain about Clustal Omega, a highly used bioinformatic tool for multiple sequence alignment. So this particular tool basically makes use of sequences including protein, DNA and RNA, which can be used for multiple sequence alignment. So here we have a set of different sequences which is obtained from P53 tumor suppressor gene for different organisms, which have been copied on the notepad. So these sequences can directly be taken and can be pasted onto the dialog box, which is available on the dashboard. So I am just copying all the sequences which are available here, and I am just pasting it onto the dashboard, which is available on the Clustal Omega website. So once it is pasted, I have already pasted them. You can choose the parameters required. I am using Clustal W with all the character counts, and I am just submitting for the results. So in this case we generally see that it takes time for the multiple sequence alignment depending upon the sequences that you have given. Now here in the result that is obtained, you can see a series of dots and you have continuous aligned sequences. Now the dot regions are basically indicating that these are distant regions, the non-conservative regions, and the continuous line sequence are the one in which you have phylogeny, or there is a close relationship between the organisms. So here this sequence will basically tell you about the different amino acids, the area of similarities and area of distance, and it also tells you about conservation, that is area of absolute similarity. So in this case you can see isoleucine and valine which are of the same color, which is telling that they are closely related to each other. So this is something which you can get from the Clustal W alignment. Apart from this, we have different parameters. One such parameter is the tool output. So in this tool output, these are basically the indicators which tell you about the similarity and the differences. As you can see from the observation here, you have regions which are marked with asterisks and you have regions which are marked with dots. So asterisks are the one which basically tell you there is close similarities, and the dot region are the one which tell you that the sequences are quite different from one another. Apart from that, we also have the phylogenetic tree construction, which is one of the major add-on to this. So the phylogram is basically the one which is giving you the degree of relationship between one sequence to the another sequence. In this case you can basically see that here the output is given based on the relative scores. So if we just zoom, we can get the idea that we have different organisms that are present in these sequences, including chimpanzee, human beings, monkey, wolf, hamster, mouse, frog and shrimp. So from this phylogenetic tree, it is very clear that chimpanzee and human beings are very closely related to each other, and the next node which immediately follows to it is that of monkey, followed by wolf, hamster, mouse, frog and shrimp. So from this it is very clear that human and chimpanzees are very closely related to each other, wherein chimpanzee and shrimp are very distantly related to each other. I hope this video has given enough information with respect to Clustal W, and I have shared the information that I have learnt from this faculty enhancement program. Thank you very much.